Hello, my name is Lillian Zhang, and my study is using Wnt1 and SOX10 as markers for craniofacial osteosarcoma tumor cells for retainment and neurocrest stem cells, and I am working with Dr. Mark Hansen at UConn School of Medicine. Osteosarcoma (OS) is the most common histological form of primary bone cancer and is the third most common cancer for adolescents. There is an estimate of 5 million cases annually for U.S. patients under the age of 20. The survival rate of primary craniofacial OS is approximately 55%. Completed surgical resection determined by margin status correlates well with survival. However, resectability is limited by the tumor spread to adjacent areas, which occurs in 50% of cases. Therefore, after reading an article provided by my mentor, I realized the need to better identify tumor margins to refine surgical resection, thus provide better patient outcomes. I am conducting research under the guidance of my mentor, Dr. Mark Hansen, in his lab at the University of Connecticut School of Medicine. Dr. Hansen taught me lab methods and techniques before experimentation, such as dilutions and labeling cells immunohistochemically with antibodies. Crest assisted me with lab procedures and handling equipment. Actual experimentation began in early December and will carry on until the end of January along with analysis. The neural crest arises at the neural plate border of the vertebrate embryo and is a multipotent collection of cells. Research in 2014 from the Hansen lab has suggested that primary craniofacial OS tumors, which are highly similar to the neurocrest stem cells that persist in the adulthood, as a group of multipotent cells. It is highly suggested that the tumors arise in parts of the skull that are derived from the embryonic rest of the neurocrest due to the stem cell's malignant transformations. I hypothesized that primary craniofacial OS tumors do arise from the neurocrest stem cells through lineage fate mapping techniques, which has been previously used by other studies. To test this, I will use lineage fate mapping to mark cells of neurocrest stem cell at distinct times of development. Wnt1 and SOX10 are the best markers since the two genes are still expressed after development. Two distinct immunoglobulins for Wnt1 and SOX10 will be used to mark cells of neurocrest origin. A fluorescent tag will be applied to each antibody, so the pseudocolor red will represent Wnt1 and pseudocolor green will represent SOX10, and the yellow color identifies that both genes are present. The groups tested are 8.5-day embryos, 10.5-day embryos, and 16.5-day embryos, in addition to humor bone tumors. The sliced tumors are viewed under a fluorescent microscope for microdissection, and statistical analysis will be completed using GraphPath Prism. Thanks for watching!